fine.
Yeah.
Okay. Can everyone on Zoom hear me from here? If you can just like give a thumbs up or yes. Okay, cool. All right. Welcome to the Bio 285 review session for topic 3. So starting off, you know that in the cell, proteins must be trafficked to specific sites inside or outside of the cell in order to function properly. For example, we need insulin must be released into the bloodstream in order to do its job. It can't stay inside of the cell. Another example would be DNA polymerase. It needs to be moved into the nucleus in order to undergo its function. And in eukaryotic cells, we use signal sequences in order to localize proteins to specific sites within the cell. So here on the slide is a few examples of different types of signaling sequences. So when proteins are made in the cytosol, they can go to either mitochondria, peroxisomes, chloroplast, or the nucleus. And then if they are ER-bound proteins, then they will enter the endomembrane system. So ribosome location is really important and usually determines where proteins will be localized. So free ribosomes that are just floating around in the cytosol, these proteins that these ribosomes produce are usually proteins that will either be localized to the nucleus, mitochondria, peroxisomes, chloroplasts, or just stay in the cytosol as is. On the other hand, membrane-bound ribosomes, so these are ribosomes that are going to be on the outside of the endoplasmic reticulum, so they're kind of going to be located on the outside of that. These proteins are going to be translated into the endoplasmic reticulum, which will then enter them into the endomembrane system. So,
Sorry.
it's okay. So basically, the difference that you want to know is that free ribosomes make proteins in the cytosol, and they will enter one of these 4 at the bottom of the screen. And the ribosomes that are usually docked on the ER will make proteins that are bound for the endomembrane system. So the endomembrane system, what is it? It includes the endoplasmic reticulum, the Golgi bodies, lysosomes. It's part of the secretory pathway and helps when trying to get proteins embedded in the membrane. So any protein that functions in any of these, either the Golgi body, ER, the lysosome, needs to be secreted or embedded in any membrane. It must have an ER localization sequence. So what are signal sequences? Signal sequences are a group of amino acids in an amino acid chain. This is part of the primary structure of a protein. Essentially, it tells proteins where its destination will be. It is not added after proteins are made. That is a key difference with signal sequences. These sequences are added as the protein is being made. So it's part of the amino acid sequence that the protein is made up of. On the other hand, localization and retention sequences. For the endomembrane system, proteins that are bound for the endomembrane system will have an ER localization sequence because they need to be localized to the ER in order to enter the endomembrane system. And retention sequences, that means if a protein needs to stay inside of the ER or the Golgi or be embedded in any cell membrane, it needs its retention sequence in order to stop along its way. Some other localization sequences include nuclear localization. That means these proteins that are made in the cytosol will be sent to the nucleus. And mitochondrial localization, surprise, surprise, goes to the mitochondria. So the endomembrane system signal sequences. So the picture on the left is, on the right, sorry. is the endomembrane system, which consists of the endoplasmic reticulum at the top right corner. These proteins that are made here will enter secretory vesicles that will travel through the cytosol and dock in the Golgi body, which is this thing in the middle. And after they are docked into the Golgi body, they will also be transported into vesicles again, which will then fuse with the cell membrane to release these proteins to the outside of the cell. So once a protein has an ER localization sequence, it is bound to the endomembrane system and will stay in it until the protein is secreted. So if it has an ER localization sequence, it's going to enter the ER and it will go all the way through until it's secreted unless told otherwise. And in order to stop anywhere along this chain or this system, it needs a retention sequence. So for example,
Okay.
so an ER retention sequence. So ER retention sequence, when a protein must function in the ER, it needs an ER retention sequence. Same with the Golgi body. So if it needs to function in the Golgi body, it needs a retention sequence for the Golgi. And if it needs to be embedded in any membrane, it needs a cell membrane retention sequence. So a question for you all. If a protein is made to replace a receptor on the cell membrane, this protein was translated by a ribosome bound to the ER. Imagine that this protein underwent a mutation where the retention sequence was deleted. Assuming that the protein's ER localization sequence is still intact, where will the protein end up after being made? You guys can either type it or just sound it out.
Okay, I raise my hand.
Yeah.
Well, if I remember correctly, we'll probably go to the Golgi,
Mhm.
and we'll probably end up like having the lysosome, cell surface, or the other. Because it wouldn't stay in the ER, it would go through
Mhm. The endomembrane
pathway.
system. Yeah.
Yeah.
Where in the system do you think it would end up though?
oh,
If-
and
so if it-
cytosol? Is it not?
Would it be excreted?
Yes, exactly. Because once you have an ER localization sequence, it's going to enter that pathway. And if it does not have a retention sequence, it's going to go all the way through until it's secreted. It needs a retention sequence in order to be stopped anywhere along that pathway.
Okay.
Yeah.
Anyways,
Uh-huh. Does that make sense too? Okay. I hope everybody on Zoom also got that.
Are there any questions? Do you want me to
Yeah.
next slide? Okay.
Okay, and last slide for me is nuclear import. So I'll just walk through this whole process. So number one, here on the bottom right corner, important will bind to its cargo protein that has an NLS sequence. If a protein has an NLS sequence, that means it's nuclear localized, so it has the ability to go into the nucleus. After important binds to this cargo protein with NLS, it will enter through a nuclear pore seen on number 2 on the diagram on the right. Once inside the nucleus, RAN will be bound to GDP, and by the help of a GEF, this will cause RAN to let go of GDP, causing GTP, or allowing GTP to bind with RAN. So once RAN is bound to GTP, RAN is rendered active. So after RAN is activated by GTP, it is allowed to bind to important. So after it binds to important, some conformational change happens and important will release this cargo protein and now is able to function inside the nucleus and do whatever it needs to do inside the nucleus. While still bound to GTP, RAN will bind to important. it will stay bound to important and exit through a nuclear pore labeled number 4 on the diagram. After the RAN important complexes in the cytosol, a gap will cleave a phosphate off of this GTP, causing it to remove a phosphate, making it into GDP. So now RAN is inactive, which will now release this important, which now can go through and go bind to more proteins as it needs. And a little tip is GEFs are usually found inside the nucleus and GAPs are found in the cytosol. So that's a good rule of thumb or a good thing to remember. GEFs are found in the nucleus, GAPs are found in the cytosol. And if a question asks if they're in somewhere where they're not supposed to be, make sure you know the difference of what they do in each place. Is this any confusing or any questions about this pathway? No?
Okay.
Do you have any questions? No? Has anyone seen a question?
No,
Okay.
okay. Nuclear export. So step 1, RAN-GEF binds to RAN GDP, and then that causes a conformational change that results in the release of GDP. And then this open binding pocket allows GTP to diffuse into RAN. And then for step 2, export in binds to RAN GTP and it undergoes another conformational shift. And then this allows it to bind to the nuclear export sequence or signal sequence or NES sequence of the cargo protein, which you can see in step 2. And then once this complex is together for step 3, it is transported out of the nucleus through the nuclear pore. And then step 4, RAN-GAP binds to RAN, which increases RAN's intrinsic catalytic ability to hydrolyze GTP to GDP. And it does this because RAN-GAP has like an arginine finger. It's just an arginine amino group, and it stimulates the active site of RAN. And then in step 5, now that RAN is bound to GDP, it undergoes a conformational shift again and releases the cargo protein into the cytosol. And you don't need to know the mechanism to which the cargo, not the cargo, export and goes back into the nuclear pore. You just need to know that it does go back in. Is there any questions about this? Okay.
I guess the question is, so GAP and GEF are always for exporting and importing.
Yes.
Mhm.
Okay. And then we went over it in both import and export, but just a more generalized slide for the roles of GEF and GAP. They're both proteins that act on the RAS family of GTPases. An important thing to note, she may ask on an exam or like even the final, she'll give proteins and ask like which are enzymes, which are not. RAN-GEF and RAN-GAP are not enzymes. They're just regulators of an enzyme. So GEF facilitates the exchange of GDP for GTP, and it does so by inducing a conformational change, which decreases the affinity of GDP, and then GTP can diffuse in. And then for GAP, it stimulates the intrinsic GTPase activity of RAN. Okay, and then a practice question. How would nuclear export be affected if there was a loss of function mutation in RAN-GAP? So step 4. That's close. It wouldn't release into the cytosol, but it wouldn't go back in with the cargo protein. It would just accumulate in the cytosol.
This whole complex. So if GAP isn't able to work here and cleave off this phosphate, this whole complex is going to stay bound together, and it's just going to stay in the cytosol. So every time this complex leaves the nucleus, it's just going to be this accumulating in the cytosol. Just keep on coming down.
And then after a while, it also prevent other proteins from being exported from the nucleus, as all the exportins would just be stuck in the cytosol. Okay, transcriptional regulation. So transcription factors regulate RNA polymerase binding to promoter DNA. For basic transcription factors, what's required for it to occur at all? Basal transcription factors in eukaryotes and sigma factors in prokaryotes. And then there are regulatory transcription factors, which either upregulate transcription, which are positive, or suppress transcription, which is negative. So like a repressor protein in like the lac operon, for example. And then another transcriptional regulation, but only in eukaryotes, is with chromatin. So chromatin packaging determines which genes can be expressed. There's euchromatin, which is where the DNA and histone complexes are loosely wound. So RNA polymerase and transcription factors can access the DNA easily. And then in heterochromatin, the DNA and histone complexes are tightly wound and packed together. So the DNA is inaccessible to the RNA polymerase and transcription factors. Okay, and then RNA processing in eukaryotes, there's the 5' GCAP, which is a covalently added guanosine nucleotide on the 5' end that's added in the opposite direction. This prevents the nucleases from chewing the mRNA or degrading it. And then there's the poly-A tail, which is just a long chain of adenine nucleotides added to the end of the mRNA. And this does get chewed by the nucleases, but the point is that it should be like long enough so it won't like in a short amount of time, but once it does reach the end of the poly-A tail, it just gets degraded. And then the poly-A tail is also required for export from the nucleus. Okay, and then there's also splicing. So in the RNA, there are introns, and these are non-coding regions, so they have to be spliced before translation can occur. The exons are the areas that do code for proteins, and the enzyme that splices these out is called the spliceosome. And then there's also alternative splicing, which allows multiple different proteins to be made from the same stretch of DNA. And just little things to note, only the middle exons can be spliced out. The first and last exons have to be retained because those are what contain the start and stop codon. And then another thing to note is that introns are always introns, so they're going to be removed no matter what, even in alternative splicing. Okay, and then here's just an overview of all the different gene expression regulation in eukaryotes. A good thing that to note, like what you should be able to do, I don't think it's on the problem set, but it can be on a future exam. You should be able to describe an example for each type. So like one for transcriptional control, which we talked about earlier, like chromatin's example. For 2, RNA processing, you could say poly-A tail. So you should have an idea of one for each of these. Okay, and then comparing gene expression in eukaryotes and prokaryotes, the similarities are that they both regulate mRNA degradation rates. They regulate mRNA localization, the rate of translation. They regulate protein activity and degradation, and they regulate transcription with transcription factors. And just at the bottom of differences, you can see that eukaryotes use the basal transcription factors and prokaryotes use the sigma factor. And then the other difference is that eukaryotes do have chromatin, whereas prokaryotes do not, as they don't have histones. And then eukaryotes have a nucleus, so mRNA is processed and transported. In prokaryotes, there's no nucleus, so things like the G prime cap, the poly A tail, that is not used in eukaryotes because they don't have to leave a nucleus, but they do have their own mechanism of RNA processing. We just don't need to know what it is for the purposes of this class. And that's all we have prepared. Does anyone have any questions?
Oh, sorry.
No,
And you can go ahead, you raise your hand first. And then see it.
It's not really so much on like a lot of the content, but I was just having a lot of trouble with the problem set. So I was wondering if we could go over some of the problem set stuff.
Yeah, we
have a
can.
question
Yeah.
About this stuff.
No, about the problems.
Yeah, we're gonna go over the problem set once everyone asks their questions on like the slideshow, because then we're just gonna stop recording and then we'll do the problem set questions. So are there any questions about like content and not the
Like,
problem set? Yeah. No, okay,
Sigma's good too.
yeah, there's nothing here. So I'll stop the recording.